Hello, biologists. So you have already finished about half of chapter 32. We're discussing animal diversity, and we're going to talk about body plans next. So if you think of some different animals, maybe a bumblebee, a cat, a sponge, a person, a frog, you probably are not going to get those confused with each other, right? It's pretty easy to tell this is a cat and this is a frog. Our bodies have very different morphologies to them. However, for all the differences in our morphologies, we do have a few set of body plans that are pretty consistent through different animals. So by just discussing a few of these plans, we can really see how a lot of animals developed. A lot of body plans have been conserved. That means through time, they have kept that same body plan. Sometimes over the course of evolution, some species have changed their body plans many times. Our body plans include asymmetrical. That means that there is no line that you could draw in the middle of the organism and have two equal halves. There are animals that are radially symmetrical. So that would be like this pot that you can cut in half here, or you can cut in half here, or you can cut in half here. And you have equal halves through multiple different lines of symmetry. Just like this anemone here. Multiple different areas where you could cut it in half and have two different mirror images. Bilateral symmetry tends to mean you have a top and a bottom, a front and a back, there a left and a right. There is one line of symmetry that you could cut this shovel down, one line of symmetry that you could go through this lobster. It's got a head and a tail that are not the same, but it has a left and a right that are more or less the same. That's what it means to be bilateral. So these three types of symmetry, where would you put people? Are we asymmetrical? Do we have radial symmetry or bilateral symmetry? So hopefully you said we have a top and a bottom, we have a head and a tail end, we have a front and a back. Those are different from each other, but our left and right are pretty similar. You could draw a line down the middle of your body, divide yourself into a left and a right, and both sides would look mostly like mirror images to the other. So in a radial symmetry, this is common in something that is sessile, which means attached to a surface, so like a barnacle attached to a boat, or something that's planktonic, like a jellyfish kind of drifting through the ocean. That radial symmetry allows it to be able to interact with its environment on all sides because it's equally likely to encounter something on any side. A bilateral organism is going to be typically an animal that's more active. Will tend to have cephalization occur. Cephalization means we have like a head that typically will have a central nervous system. We may have sensory organs here, so like eyes, mouth, nose, ears, things to help us interact with the world around us. In an organism with bilateral symmetry, we have a distinct head and a distinct tail. The head end is the anterior, the tail end is posterior. We also have a distinct back and a distinct belly. The back end is your dorsal side and your belly end is your ventral side. So if you've heard of like a dorsal fin, that's a fin on the back of a marine animal. If you've heard of, put your posterior in the seat, they're saying put your booty in the seat. These are 4 words that you need to be very comfortable with. Anterior, posterior, dorsal, and ventral. Those words will show up on exams and quizzes. They will show up in lab. They will show up the rest of the semester. Anterior is your head. Posterior is your tail end, your booty end. Dorsal is your back. Ventral is your belly. Okay, body plans are also characterized by different organizations of tissues. As we already said in our first video, tissues are collections of specialized cells that act as a functional unit. So you could have blood tissue, bone tissue, epithelial tissue, cartilage tissue, adipose tissue, different types of tissues that we have in animals. Although sponges do lack true tissues, they are animals without true tissues. Most animals are made of tissues. And in animals, we start developing different types of tissues when gastrulation is happening. If you remember, we start to have that pouch come in, different layers of cells happen. These different layers are called germ layers, and each germ layer is going to develop into different types of organs. So you can see ectoderm forms the exoskeleton, mesoderm forms the organs, endoderm forms the inner lining of organs. So our germ layers then that we have ectoderm, endoderm. We talked about these two already in the first lecture video. Ectoderm is the outer surface of the embryo, and it is going to become the skin and the nervous system. Endoderm is the inner layer. Here it's in the yellow, and it's going to become the lining of your various organs. So it's going to line your digestive tract. It's going to line your respiratory tract. Mesoderm, we haven't talked about yet. It's unique to some animals, but not all animals have a mesoderm. It's I think of M for meso, M for middle. The mesoderm is the middle layer. And this will become your organs, your muscles, and your connective tissues. Some animals, as we develop, are diploblastic. These are animals that only have an ectoderm and an endoderm. Some animals are triploblastic. They have all three germ layers, an ectoderm, endoderm, and mesoderm. Most triploblastic animals, most animals with all three layers, are going to have a coelom. A coelom is a hollow space, a body cavity within the animal, usually filled with fluid or air, and it is going to separate the digestive tract from the outer body wall. In a eucoelomate, there is a true coelom. That means that it is covered with mesoderm, both on the outer lining of the body cavity and on the inner lining of the cavity. The body cavity, that coelom, is completely surrounded by mesoderm. Pseudocoelomates, so pseudo means false. You have a body cavity, a coelom, that is lined with mesoderm, the red here on the outside of it, but there's no mesoderm on the inside of the cavity. It's just endoderm. However, even though it's called a false coelom, it still acts exactly the same as any other type of coelom. And then there are acoelomates that don't have a body cavity at all. You can see there's no sort of cavity in here. It's just solid. So what is the difference between a eucoelomate and a pseudocoelomate? The purpose of your body cavity, there's multiple purposes of it. One is to cushion the organs. So the organs that are suspended within your body are kind of cushioned and protected within that fluid. In something that's soft-bodied, so something like this worm here, that fluid, it's not compressible, so it kind of acts as a sort of like skeleton that it can kind of let its muscles work against. The cavity also lets internal organs grow and move independently of the outer body wall. So you can't see every beat of your heart because of that cavity around it. Although your chest may rise and fall when you breathe, you can't actually see, the lungs moving because of that pleural cavity that you have that is derived from our coelom. Okay, the last thing we're going to talk about in body plans are protostomes and deuterostomes. So if you remember back to the gastrulation, how you develop this indentation that becomes a blastopore, and then the blastopore is going to keep growing through and create a second hole at the other end. So we have these two organisms, one is a protostome and one is a deuterostome. Both of them have developed that opening all the way through both ends. So the difference between this protostome and the deuterostome, we're going to see differences in how the coelom is formed, what's going on with the blastopore, and we're also going to see differences in how cleavage occurs. So first, let's look at how the blastopore is different in protostomes and deuterostomes. So as the blastopore has formed from one opening, developed on through to the other opening, in a protostome, that first opening is the mouth. So proto means first. Think it's like a prototype. Proto means first, and stome means mouth. So first is the mouth. Then as it forms through the other end becomes the anus, and now you have this digestive tract. In a deuterostome, the first opening, that blastopore, becomes the anus, and then as it pushes on through, the second opening becomes the mouth. So deutero means second, so second opening is the mouth. That's one difference between a protostome and a deuterostome. So here's a question for you. Soon after the coelom begins to form, a researcher injects a dye into the coelom of a deuterostome embryo. Initially, the dye should flow directly into what? The blastopore, blastocele, archenteron, or pseudocoelom. Next difference between a protostome and a deuterostome is spiral cleavage or radial cleavage. Here in a protostome, we have what's called spiral cleavage. So these planes of cell division are diagonal to each other. See how they don't line up? They're diagonal to each other. Also here, cleavage is determinate. That means each of these cells knows exactly what it is supposed to turn into. So if you took one away, you will end up with an organism missing some of its body. And that one that came away wouldn't develop into a whole individual. In deuterostomes, we have radial cleavage. So you can see how the cells kind of nicely stack on top of each other. Cleavage is also indeterminate. So each of these cells still could develop into anything in the body. So if you took one of these cells away, that cell could turn into a whole new organism, and the 7 that were left behind could still develop into a whole organism. They are capable of forming identical twins. So this could divide in half, and it would be just fine. It has not yet determined what each cell needs to turn into. It is indeterminate cleavage at that 8-cell stage. Another way that protostomes and deuterostomes differ from each other are in coelom formation. So again, remember during gastrulation, we form this blind pouch called the archenteron. In a protostome, the coelom is going to start forming at the same time. In a protostome, you'll have these solid masses here of mesoderm that split on each side. This is called schizoccelous development, and that coelom will form around the digestive tract here. In deuterostomes, you have enterocoelous development. So the coelom here, it isn't moving over here. The coelom is kind of just budding off of this archenteron, and then it'll form the coelom around the digestive tract. Okay, so protostome development includes, is it A, radial cleavage, B, determinate cleavage, C, a diploblastic embryo, D, the blastopore becomes the anus, or E, the archenteron is absent. And protostome characteristics include, is it A, a mouth that develops secondarily far away from the blastopore, B, radial body symmetry, C, radial cleavage, D, determinate cleavage, or E, absence of a body cavity. Okay, so we are going to pick up in our next video, finishing this chapter, discussing how animals have diversified. Again, let me know if you have any questions. I have office hours that are free for you to come in, and I have an email that I will answer as soon as possible. I will talk to you later. See you. Bye.